Hi guys, this is the skeletal muscle physiology lesson. This is a new lesson and you are responsible for all the material, and there will be questions directly from it. It is not review. However, I will tell you that a lot of this will be sort of repeated from the cardiovascular block because there's a lot of overlap between the physiology of cardiac muscle and skeletal muscle. Nevertheless, you are responsible for all of it. I think it'll be a very useful review for you. You should, as I go through this, pay attention to the differences between skeletal muscle and cardiac muscle. It is a good traditional way to teach this material, to contrast the two, and I will be doing that as I go through. And you are responsible for knowing the differences as I bring them up. Okay? Just by a quick introduction here. Again, this is my contact information. Here are my office hours. I'm available on Sunday. I'm appearing. Of course, you can always meet with me by appointment. And do note that this video is meant to go along with the course notes that I've given you. And if you wish, you can just sit back and watch the video, make the occasional annotation to the course notes, and you should be good. It's designed so that you really don't even have to take notes. However, if you do not like that idea, you don't want to have to read through the notes or look at them, you think I'm a blowhard academic and I go into too much detail, don't do it. Just watch the video, take notes on the slides, you'll be fine. All the content is there. And in contrast, if you want to just read the notes and you don't want to watch me sit here and flail my hands around, fine, read the notes, it's all there. Okay, any way you want it, that's the way you got it. Do whatever you wish. Each is sufficient on its own, or you can just do it the way that they're designed and watch and follow along. Okay. Again, we're talking about skeletal muscle. The first part of this will be pretty quick because I think it's review from your histology. But nevertheless, I want to go through it to make sure we're all on the same page. So this is a skeletal muscle here, and individual skeletal, and these skeletal muscles, well, muscle in general makes up 50% of your total body weight. Most of that is skeletal muscle, of course. The individual cells in these skeletal muscles are called muscle fibers, all right? Skeletal muscle cells are muscle fibers. They're anywhere from 10 to 100 microns in diameter, and they are, in terms of length, the entire length of the muscle. So this is different from cardiac muscle, where the length was 100 microns, and they were about 10 or 15 or 20 microns in diameter. And then they were all hooked together in a functional syncytium at the gap junctions, at the intercalated discs. This is different. You have one cell running the length of the muscle, all right? And that is a muscle fiber. The muscle fibers have myofibrils attached, have myofibrils. Bundled connective tissue makes tendons. So this is surrounded by connective tissue. You guys know this from anatomy. It sort of coalesces at the end of the muscles to make the tendons that you see. And now we have individual muscle cells have myofibrils inside them, and these are bundles of actin and myosin and troponin and tropomyosin that again run the entire length of the cell. They are made up of individual sarcomeres, very similar to cardiac muscle cells. You have sarcomeres, and the sarcomeres have thick filaments, which are myosin, and they have thin filaments, which are actin, tropomyosin, and troponin. Okay, very similar to cardiac muscle. Okay, and as you can see, these overlap, and this is where the contraction takes place. So we have areas that are just actin, we have areas that are just myosin, and we have areas of overlap. Okay. And you can see those areas there on the electron micrograph at the top here. And again, this is just a very quick review because I know you've had it. The A band is basically just a myosin. It's the heavy filament, all right? So the A band runs from the tip of the heavy filament to the tip of the heavy filament. The I band is actin, but it's the part of the actin that does not overlap with the heavy filament, okay? So it's just the light zone here. And of course there is actin that overlaps with the myosin here. It's the thin filament, excuse me. And there is thin filament here that overlaps with thick filament, but the I band does not include that. You have the H band or the H zone, which is in the middle of the A band, and it is the portion of the A band which is just myosin with no actin, okay? So this is myosin and actin overlap. This is just myosin here in the H zone. And then you have your Z line, which is a line of proteins here to which actin can anchor. So you have actin on this side anchoring and actin on this side anchoring. At the Z line, it holds the actin in place. And then you have the M line, which runs through the middle of the A band. It actually runs through the middle of the H zone. And these are proteins to which the myosin anchors, and it holds the myosin in place, okay? All right, so that was really quick, and now this is a very quick review of the neuromuscular junction, which I know you guys just had a lecture on, but, and I had an overview slide that I actually cut out because it wasn't really necessary, but I showed that to you in the synapse lecture. But that just showed the same thing that you're seeing here. You have your action potential coming out of the spinal cord. Your action potential is propagating. It hits the nerve terminus here. This is the presynaptic membrane. It depolarizes the membrane. It allows calcium to come in through N-type calcium channels. That causes the vesicles with the neurotransmitter, in this case acetylcholine, to fuse with the presynaptic membrane. You get exocytosis of acetylcholine into the neuromuscular junction, into the synapse, and it diffuses across to the postsynaptic membrane, which is the skeletal muscle. And you have your synaptic folds here. At the top of the folds, you have your acetylcholine receptors, which are also the channels. Acetylcholine binds, opens the channels, it causes depolarization of the membrane. You get an end plate potential that every time in a healthy muscle that hits threshold, it causes an action potential to be generated, and your action potential propagates away from the synapse. Okay, the bottom of your synaptic folds has the acetylcholinesterase, which eliminates the acetylcholine, and when this release stops, this eliminates the acetylcholine, and your acetylcholine goes away, and that stops the stimulation. Okay, so that was your quick neuromuscular junction. This is an electron micrograph of it. You guys remember that, all right? Your action potential propagates away, and it dips down into the T-tubule, very similar to cardiac muscle. You have these invaginations of sarcolemma, cell membrane, which dip down into the fiber, okay? And your action potential propagates down into these T-tubules. And those are depicted here in these two diagrams, here, the red, and here. Okay, and those are contiguous with the cell membrane, the sarcolemma, and your action potential propagates down in here. And notice that whatever's inside this T-tubule is basically extracellular space, which means it has a lot of calcium in it. So what you're really doing is you're bringing calcium down into the middle of the muscle fiber. So if calcium had to diffuse from the outside of the fiber into the middle, that would be a really long distance to diffuse, and it would take a very long time. So your T-tubules bring your calcium into the middle of the fiber where it can be released, where it can cause calcium release from the sarcoplasmic reticulum, which is right here. And that's like right on top of the myofomus. So it really, really reduces the distance of diffusion for calcium, and it causes a very quick contraction as opposed to a really, really slow contraction, which is what you get if the calcium had to diffuse in from the outside of the fiber. Notice that the sarcoplasmic reticulum in skeletal muscle has these lateral sacs. Those come in very close to the T-tubule. So we have them, they're called lateral cisternae on this slide. And where they come in very close to the T-tubule, you have what are called TSR junctions, okay? And that's this right here in a resting muscle, all right? And your TSR junctions have these proteins in apposition. It's a very, very narrow junction, just like it is in cardiac muscle. You have these voltage sensors or depolarization sensors in the sarcolemma of the T-tubule. These are also called DHPRs for dihydropyridine receptor. They bind calcium channel blockers, and the reason is because they are actually calcium channels. Unlike cardiac muscle, these calcium channels conduct very little calcium, and they do it extremely slowly, so they do not bring in significant amounts of calcium. Okay, they will do it, and it is measurable, but this will be the last you hear me mention it because it's irrelevant. Unlike cardiac muscle, where it's necessary for contraction, the calcium influx here is irrelevant. And what this sensor is actually doing is it's changing configuration in response to voltage, and I'll go through that in a minute. Opposed to those are your ryanodine receptors or your calcium release channels. This is the same as cardiac muscle. There are different type of ryanodine receptor, but nevertheless, they act very similarly, okay? And they are SR calcium release channels. You do get calcium release through them. So the way that this works is different from the way that it works in cardiac muscle. In cardiac muscle, an action potential propagated down the T-tubule. It opened up this calcium channel, calcium diffused into the space, it binded to the ryanodine receptor or the SR calcium release channel, and you got release of calcium. It's different for skeletal muscle. Skeletal muscle will operate in the absence of calcium in the extracellular space, and that can be proven in a laboratory. Okay? Instead, the mechanism is what we call a mechanical mechanism. And what that means is that these two proteins are physically attached. They're bound to one another. And that's the current theory, and pretty much everyone accepts it. So the action potential propagates down the T-tubule, and now it causes this voltage sensor to change configuration. And it changes its conformation, and because it's bound to the ryanodine receptor, it can cause the ryanodine receptor to change its configuration and open and release calcium. And that happens in the absence of extracellular calcium. You just need calcium in the SR. And the vast majority of the calcium that causes contraction of skeletal muscle, virtually all of it comes from the SR, okay? And that's all you need. So that's different from cardiac muscle, and you should note that. So it's not CICR, where calcium-induced calcium release takes place here. The primary mechanism is, at least in terms of the way that the release is initiated, is a mechanical mechanism where the two are attached. So in summary, we have our action potential propagating down our nerve. Your ACH finds your end plate and causes an end plate potential, which causes the membrane hit threshold, which means you fire an action potential. The action potential propagates down through the sarcolemma into the T-tubule in the T system, and that causes calcium release from the SR through this mechanical mechanism. All right? So that is the end of video 1 in this lesson, and in video 2 we'll talk a little bit about how the actin and myosin act to cause contraction.